All right, so tonight's session, we'll take a look at the final chapter in our textbook, taking a look at different types of metabolism and really one of the key forms of energy that you come across, ATP and such like that. So we'll start by looking at some general ideas behind metabolism and ATP energy. So ATP, the energy storage molecule, links energy producing reactions with energy requiring reactions in cells. So as you'll see during the course of our slides tonight, you have some reactions that are defined as catabolic reactions. And in a catabolic reaction, energy is going to be produced, which essentially means that energy is a product of that reaction. Whereas other reactions that you come across are defined as being anabolic reactions. Now, in those cases, energy is required to be put in order for that reaction to occur. And in a sense, therefore, essentially, energy is a reactant in that process. So in terms of energy, the main source of energy that is used to have anabolic reactions take place is your ATP. And we'll get into the structure of ATP as we go along. When ATP energy is used from ATP, you essentially have the ATP converted into ADP, essentially like adenosine triphosphate, adenosine diphosphate, essentially the loss of a phosphate group. And then essentially when energy is produced, then you regenerate the ATP and such like that. So you'll see that as we go through our slides this evening. All right, so metabolism. Metabolism involves all chemical reactions that provide energy and substances needed for growth. Okay. Catabolic reactions. Now, essentially, we saw that catabolic reactions are energy-producing reactions. So another way to sort of look at that is that catabolic reactions break down large, complex molecules to smaller molecules. And by doing so, they have energy that essentially is released. They provide energy. So for catabolic reaction, you have the breaking down of larger complex molecules into smaller molecules. Now, essentially, anabolic reactions really are, in a sense, the opposite of that. They use energy, essentially in the form of ATP, to build larger molecules. So your anabolic reactions sort of have smaller molecules that combine to build larger molecules. And when you have larger molecules that break down to smaller molecules, energy is released. But when you need to build larger molecules from smaller ones, then you have to put energy in. And again, essentially that energy is going to come from your ATP. Okay. So stages of metabolism. So catabolic reactions are organized in stages. First one, stage one, digestion and hydrolysis. Of course, what's hydrolysis? Hydrolysis essentially usually is the splitting apart upon reaction with water. So they break down large molecules to smaller ones that enter the bloodstream. So that's stage one, digestion and hydrolysis. Stage two is degradation. Now, degradation breaks down molecules to 2 and 3 carbon compounds. So it's still sort of a catabolic type of process because you're still sort of breaking down larger molecules into smaller molecules. When we get into carbohydrates, we'll see that digestion and hydrolysis essentially break down your polysaccharides and your disaccharides into your monosaccharides. And then the degradation process essentially takes those monosaccharides, which I believe is usually glucose, and breaks that down into smaller 2 and 3 carbon compounds. So it's still the process of breaking down smaller compounds, larger compounds into smaller compounds. And then oxidation, which would be stage 3. Now remember, oxidation essentially is an increase in the number of carbon to oxygen bonds in the organic chemistry world. The other definition of oxidation, which comes into play here a little bit more, is that oxidation, and we saw this in the earlier chapters, is the loss of electrons. The acronym oil rig, oxidation is loss, reduction is gain. So the oxidation of small molecules in what is known as the citric acid cycle and electron transport provides ATP energy. So in the 3 stages of metabolism, Large molecules from foods are digested and degraded to give smaller molecules that can be oxidized to produce energy. So really the first two stages are the breaking down part, larger into smaller, and then the oxidation part is when those smaller molecules are oxidized to produce energy. All right. So essentially what you have here is, I just want to see if they give a little bit of a larger picture of it, but they don't. So what you have on the slide here is sort of an outline of the stages of catabolism, catabolic reactions, for essentially three main types of foods that we take in and digest: proteins, polysaccharides, and lipids. All right. So essentially, and we'll go through this in more detail, digestion and hydrolysis, stage one. Proteins become amino acids, polysaccharides become monosaccharides, lipids become fatty acids. Okay. And we'll see in the slides how the cell membrane is involved in this. But, if you think about what we talked about in, chapters 13, 15, and 16, we know that proteins are made up of amino acids. Polysaccharides are made up polysaccharides are made up of monosaccharides, and lipids are made up of fatty acids, larger to smaller. Then stage two, degradation and some oxidation to smaller molecules. So here's sort of where the paths sort of converge, or the three different processes converge. Because in most instances, what you're going to generate from that is what is known as acetyl coenzyme A, which is abbreviated as acetyl-CoA. Now, in some cases with proteins, you would directly have them go into stage three, they would enter the citric acid cycle. But most of the time, you get the formation of acetyl coenzyme A, and then from there, you enter what is known as the citric acid cycle. Essentially, from the citric acid cycle, we'll talk about how we're going to produce NADH and FADH2, and then through electron transport and oxidative phosphorylation, we're going to generate ATP. So this is sort of an overall schematic of how the catabolism, or the digestion, in a sense, the breakdown of proteins, polysaccharides, and lipids work. Okay. So cell structure and metabolism. So here you just have a slide of some of the major components of a typical animal cell. All right, so you've got the nucleus, the ribosomes, the cell membrane, the cytosol, the cytoplasm, the mitochondria, then you have a blow up of the mitochondria, where you have the outer membrane, the inner membrane, the intermembrane space, and the matrix. I wouldn't go crazy about this. A lot of biology here, not so much, in my perspective, chemistry and such like that. All right. But let's just go over their cell components and function. All right. So the cell membrane separates the contents of a cell from the external environment and contains structures that communicate with other cells. The cytoplasm consists of the cellular contents between the cell membrane and nucleus. The cytosol, the fluid part of the cytoplasm that contains enzymes for many of the cell's chemical reactions. The mitochondrion contains the structures for the synthesis of ATP from energy-releasing reactions. The nucleus, which contains genetic information for the replication of DNA and the synthesis of protein. And then the ribosomes, of course, which we saw in the previous chapter, are the sites of protein synthesis using messenger RNA templates. So ATP, adenosine triphosphate. If you think about in chapter 17, we talked about essentially AMP, adenosine monophosphate, and DAMP, deoxyadenosine monophosphate. And we talked about that as a nucleotide. Essentially, the starting point for adenosine triphosphate is really adenosine monophosphate. Now, it's drawn a little bit differently here than what we saw in the previous chapter, because in the previous chapter, we had the bond to the nitrogenous base on the right and the bond to the phosphate group on the left. Here they have it a little bit differently drawn, but really it's exactly the same thing. Adenine, and if we're looking at adenosine triphosphate, that means the sugar, of course, has to be ribose and not deoxyribose. So adenine, ribose, phosphate. And that would give you adenosine monophosphate. But you can link phosphate groups to each other. You put a second phosphate group on there, you have adenosine diphosphate. You put a third phosphate group on there, you have adenosine triphosphate. Now, as you saw earlier on, right back at the beginning, essentially, energy requiring reactions take ATP in and generate ADP. And this PI essentially is a cleaved phosphate. Essentially, the energy comes from the fact that your phosphate bonds are high energy. So if you break them, there's going to be energy that's present. And that energy is going to be used to essentially drive an anabolic reaction. The process of sort of breaking apart the phosphate groups does fall under the category of hydrolysis. So every time we contract muscles, move substances across cellular membranes, send nerve signals, or synthesize an enzyme, we use energy from ATP hydrolysis. The hydrolysis of ATP to ADP, adenosine diphosphate, releases 7.3 kilocalories per mole of ATP. Okay, so or 7.3 kilocalories per mole of ATP. So in other words, for every 1 mole of ATP, the energy that's going to be released when it hydrolyzes into ADP and phosphate is 7.3 kilocalories per mole. So if you got 2 moles of ATP, double that number. You got 36 moles of ATP, multiply that number by 36. Now, ADP can also hydrolyze to form adenosine monophosphate and phosphate itself. And if that occurs, that process is also going to release 7.3 kilocalories per mole of ADP. So 1 mole of ADP will release 7.3 kilocalories per mole, 2 moles, multiply that by 2, so forth and so on. All right. So ATP, the energy storage molecule, as we saw before, links energy producing reactions with energy requiring reactions in the cells. So basically, this is the same slide as before, but now they're filling in the energy values. Okay. So essentially, anabolic reactions requiring energy. The energy that's going to be used essentially is going to be 7.3 kilocalories per mole. Catabolic reactions, energy is going to be released, okay, 7.3 kilocalories per mole. The reason why I think they're using the signs here of positive and negative is that in this case, the ATP is essentially releasing energy, the negative sign. And in this process, the ADP is absorbing energy to reform ATP. Because, you know, we've talked about like energy release, you know, sort of like a negative value, exothermic process, you know, endothermic energy being absorbed, a positive value. So, you know, I think that's why the negative and positive signs are here, because they're talking about the ATP releasing this amount of energy to be used by the anabolic reaction. And then they're talking about the ADP absorbing this amount of energy that's being released by the catabolic reaction to become ATP again. All right, all right. So let's go on to part 2. Okay, so let's talk more about the digestion of foods. So in stage 1 of catabolism, foods undergo digestion, a process that converts large molecules to smaller ones that can be absorbed by the body. We'll start with the digestion of carbohydrates. So enzymes produced in the salivary glands hydrolyze some of the alpha glycosidic bonds in amylose and amylopectin, producing smaller polysaccharides such as maltose, glucose, and dextrins, which may contain 3 to 8 glucose units. Now, glucose is not a polysaccharide. Of course, glucose is a monosaccharide, and maltose is a disaccharide. And then the dextrins would probably be polysaccharides. So, you know, I don't know necessarily if we want to say producing smaller polysaccharides, but essentially we would produce smaller carbohydrate units at this point from the amylose and the amylopectin. Now, after being swallowed, the partially digested starches enter the acidic environment of the stomach. Amylose and amylopectin are considered starches, where the low pH stops carbohydrate digestion. In the small intestine, which has a pH of about 8, enzymes produced in the pancreas hydrolyze the remaining dextrins to maltose and glucose. Enzymes produced in the mucosal cells that line the small intestine hydrolyze maltose as well as lactose and sucrose into monosaccharides. Monosaccharides are then absorbed through the intestinal wall to the bloodstream and carried to the liver, where any fructose and galactose are converted to glucose. So one of the things to sort of think about is that by the time your carbohydrates have been digested, in terms of stage 1, they're all going to be converted to glucose. Then there's going to be different carbohydrates that form along the way. Maltose, dextrins, lactose, sucrose, even fructose, galactose. But essentially at the end of the day, they're all converted into glucose. And really that's the end result of the digestion process for carbohydrates. So carbohydrates begin digestion in the mouth, lipids in the small intestine, and proteins in the stomach and the small intestine. So in stage 1 of catabolism, the digestion of carbohydrates begins in the mouth and is completed in the small intestine. All right, so polysaccharides, we eat them. Enzymes, salivary amylase, right? So salivary glands, enzyme present. It's going to break down polysaccharides into dextrins, maltose, and glucose. It's going to pass through the stomach into the small intestine. Into the small intestine, dextrins are going to be broken down into maltose. Maltose will be split apart upon reaction with maltase, right? Notice the enzyme names similar to the substrate, into glucose and glucose. Now, also present in the small intestine might be lactose and sucrose. If lactose is present, lactase will break that down into galactose and glucose. And then if sucrose is present, that will be broken down by the enzyme sucrase into fructose and glucose. And then essentially through the bloodstream and through the cells. But at the end of the day, and I'm sure we'll see this, okay, that essentially when the liver, and carried to the liver, where any fructose and glucose are converted into, fructose and galactose are converted into glucose. Okay? So I'm sure we'll see that at some point. Okay. Digestion of fats. So the digestion of fats, which of course are your triacylglycerols, begins in the small intestine, where bile salts break fat globules into small particles called micelles. The triacylglycerols are hydrolyzed in the small intestine and reformed in the intestinal lining, where they bind to proteins for transport through the lymphatic system and bloodstream to the cells. So you've got your triacylglycerol. In the small intestine, enzyme known as pancreatic lipase will break down the triacylglycerol into what is known as 2-monoacylglycerol, because essentially you're going to have hydrolysis at 2 of the ester bonds to cleave off 2 of the fatty acids, okay, leaving you with this structure, which is known as a 2-monoacylglycerol. Into the intestinal wall, where you have your monoacylglycerols and your fatty acids actually recombine into triacylglycerols, combined with protein in the form of lipoproteins. Because remember, we talked about in the lipid chapter, how are lipids transported through the body? They're transported through the body as part of these spherical particles known as lipoproteins. Through the lymphatic system, through the bloodstream, and into the cells, where the triacylglycerols will be broken down into glycerol and fatty acids. Digestion of proteins. So the digestion of proteins begins in the stomach, where HCl at pH 2 denatures proteins and activates enzymes such as pepsin to hydrolyze peptide bonds. Remember, peptide bonds are amide bonds, and we know that amides can undergo hydrolysis. Moves out of the stomach to the small intestine, where trypsin and chymotrypsin hydrolyze the polypeptides to amino acids. Ends as amino acids are absorbed through the intestinal walls and enter the bloodstream for transport to the cells. All right. So proteins are hydrolyzed in the stomach and the small intestine. So proteins, enzyme pepsin will break them down into polypeptides. Polypeptides will go from the stomach into the small intestine, where the polypeptides will be broken down further into amino acids by enzymes such as trypsin and chymotrypsin. And then the amino acids will go through the intestinal wall, into the bloodstream, and into the cells. All right. Coenzymes and metabolic pathways. So the NAD plus coenzyme is required for metabolic reactions that produce carbon to oxygen, C double bond O carbonyl groups, C double bond O double bonds, such as in the oxidation of alcohols to aldehydes and ketones. So here's an example, ethanol, ethyl alcohol with NAD plus in the presence of alcohol dehydrogenase, the ethanol being oxidized to ethanal, and then the NADH essentially being reduced, the NAD plus, essentially being reduced to NADH. In the sense, remember, oxidation, usually an increase in the number of carbon to oxygen bonds. Reduction, usually an increase in the number of carbon to hydrogen bonds. Okay, so oxidation and reduction. Oxidation reactions involve a loss of hydrogen, a loss of electrons, or an increase in the number of bonds to oxygen. Reduction reactions involve the gain of hydrogen ions and electrons, or a decrease in the number of bonds to oxygen. Metabolic pathways, oxidation and reduction. So again, just sort of a characteristics of oxidation, reduction, and metabolic pathways. The key thing that was not mentioned yet, just to make note of, is on the bottom here. Oxidation reactions generally result in a release of energy, whereas reduction reactions generally result in an input of energy. So that's sort of the one thing that wasn't mentioned in the previous sort of discussions about oxidation versus reduction. So oxidation reactions in metabolic pathways result in a release of energy. Reduction reactions result in input of energy. So coenzyme NAD+. So NAD+, nicotinamide adenosine dinucleotide, is an important coenzyme in which the B3 vitamin niacin provides the nicotinamide group, which is bonded to ADP. So essentially, here's your ADP portion, okay? Then you have ribose, okay? And then you have your nicotinamide from niacin. This is considered the oxidized form because in the ring here, you have sort of alternating double and single bonds throughout the ring. The reduced form of NAD plus is NADH. And what happens is that essentially you get sort of a reaction where you add hydrogen ions and essentially electrons to make these bonds here between hydrogen and, to make one bond between the carbon and the hydrogen, because this carbon already has a hydrogen that's present. That's this hydrogen here. So essentially you're adding one carbon to hydrogen bond, and then the other hydrogen still exists as H plus associated with it. But this is a reduced form. The NADH is the reduced form because you're adding a carbon to hydrogen bond to the structure. NAD plus participates in reactions that produce a carbon to oxygen double bond. So again, we saw this example earlier, the oxidation of ethanol into ethanal using NAD plus, which the alcohol is oxidized, but the NAD plus is reduced. Okay. Another coenzyme we come across is what is known as FAD. FAD stands for flavin adenine dinucleotide, and it's a coenzyme that contains ADP and riboflavin, which is vitamin B2, and is reduced to FADH2 when flavin accepts 2 protons and 2 electrons. So here you've got the ADP, nitrogenous base, sugar, the 2 phosphates. Then essentially the riboflavin part is the structure here, which is known as ribitol, connected to flavin. which is this sort of three-ring fuse system at the top. This is the oxidized form. In the reduced form, this nitrogen here and this nitrogen here protonate. So usually reduction, you talk about an increase in the number of carbon to hydrogen bonds. You increase the number of nitrogen-hydrogen bonds, essentially that would signify reduction as well. So this would be the reduced form, what is known as FADH2. The coenzyme FAD participates in reactions that convert carbon-to-carbon single bonds to carbon-to-carbon double bonds. So here you have what is known as succinate. And if we reacted with FAD in the presence of succinate dehydrogenase, essentially what's happening is we're removing one hydrogen from each of the two middle carbons of the succinate. Where those hydrogens end up, they end up in the reduced form of the FADH, right? Notice how the two hydrogens are here. And what happens is that that single bond between the carbons becomes a double bond. What you'll see is that in the citric acid cycle, FAD is utilized in the conversion of the carbon-to-carbon single bond in succinate to a double bond in what is known as fumarate. So this process here is something we're going to see again in the citric acid cycle. Structure of coenzyme A. So coenzyme A, which is often abbreviated as CoA, contains pantenothenic acid, vitamin B5, phosphorylated ADP, and aminoethane thiol. All right, a lot going on. So essentially, aminoethane thiol group SH. Here's your ethane part. Here's your amino part. Pantothenic acid, okay, has essentially this particular structure over here. What's missing from here would be the OH of the carboxyl group. That essentially would be used to form the amide bond. And then you have essentially this phosphorylated ADP, where you have this phosphate group at essentially, and this is drawn more like we're used to it, at the 3' position. You have this phosphorylated, a phosphate group at the 3' position. That's the phosphorylated ADP. So this is the structure of coenzyme A. And basically, usually they just call it CoA. That's how you just usually see it. So an important function of coenzyme A is to prepare small acyl groups represented by the letter A in the name, such as acetyl, for reactions with enzymes. So when you talk about what is known as an acetyl group is the following. You have CH3 connected to C double bond O, which then that C double bond O carbon is usually connected to something else. But this 2-carbon group right here, this is what is known as the acetyl group. Now, as we'll see, when you react that with coenzyme A, you actually get the formation of what is known as a thioester, a C double bond O connected to sulfur. And this structure here, which we saw earlier on in our discussions, going to be in that metabolism process, this is acetyl coenzyme A. So the sulfur essentially of the thiol group right here would then have this entire structure linked to it as well. So notice they just call it CoA because they don't want to draw that entire structure out. But that's really what you would have. You'd have the sulfur, and then you'd have the entire rest of the structure. Okay? So the thiol group SH bonds to a 2-carbon acetyl group to produce the energy-rich thioester, acetyl coenzyme A. Okay, so we talked about the fact that essentially in stage 1 of catabolism of your polysaccharides, eventually at the end of the day, they're all going to be converted to glucose. But then what happens to the glucose? Well, we're going to talk about what is known as glycolysis. So major sources of energy for the body are glucose, produced when we digest the carbohydrates in our food, and glycogen, a polysaccharide stored in the liver and skeletal muscle. Okay. Glycolysis. Glycolysis is a metabolic pathway that uses glucose, a digestion product from carbohydrates. it degrades 6 carbon glucose molecules to 3 carbon pyruvate molecules. So remember back when we talked a little bit about stage 2 degradation, and we talked about the breaking down of larger molecules into 2 and 3 carbon molecules? Well, essentially in glycolysis, glucose molecules, which contain 6 carbons total, are broken down into 3 carbon, what are known as pyruvate molecules. This takes place in the cytoplasm of the cell, and it's what is known as an anaerobic process, which means that no oxygen is required for this process to occur. If you're familiar with aerobic processes, they require oxygen to occur. An anaerobic process does not require oxygen to occur. Okay. So, a lot going on in glycolysis, and you don't have to memorize everything that's here, not at all. So in reactions 1 to 5 of glycolysis, so conversion of glucose into glucose 6-phosphate and fructose 6-phosphate, and then fructose 1,6-bisphosphate, and then essentially those conversions as well into glyceraldehyde 3-phosphate. Energy is required to add phosphate groups to glucose. Glucose is converted to 2,3 carbon molecules. And those 2,3 carbon molecules are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Reaction 1, phosphorylation. A phosphate from ATP is added to glucose, forming glucose 6-phosphate and ADP. So here's your glucose, here's your phosphate group being added. Reaction 2, isomerization. The glucose 6-phosphate, the aldose from reaction 1, because remember, essentially glucose is an aldose, undergoes isomerization to fructose 6-phosphate, which is a ketose. So glucose 6-phosphate into fructose 6-phosphate. Reaction 3, phosphorylation. Fructose 6-phosphate reacts with a second ATP, adding a second phosphate to the molecule, fructose 1,6-bisphosphate. Reaction 4, cleavage. Fructose 1,6-bisphosphate is split into 2,3 carbon phosphate isomers, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now, the fun part's going to be, where does it split apart? So, it looks like it would split apart. CH2, sorry. CH2O, this would be the carbonyl, CH2OH. So I think one portion would be this here, giving us that. And then the second portion aldehyde CH2OH. It's interesting. Where does that actually split up? Or does it split apart? So that wouldn't give a ketone. Where does that bad boy split apart? Interesting. It's interesting that the ring structure into 2,3 carbon phosphate isomers, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. It'd be nice if they showed the P's in different colors. All right, something I'll have to think about a little bit more. And then reaction 5, isomerization. Because dihydroxyacetone phosphate is a ketone, it cannot react further. It undergoes isomerization to provide a second molecule of glyceraldehyde 3-phosphate, which can be oxidized. Now, all 6 carbon atoms from glucose are contained in 2 identical triose phosphates. And trying to see, that would be this structure here. dihydroxyacetone. That would be this structure. So this here, the dihydroxyacetone phosphate, that would be this. Because acetone is CH3, C double bond O CH3. And then the glyceraldehyde, because this would have to be the aldehyde group, would be that structure there. And we know that ketones cannot undergo oxidation, but aldehydes do. So the dihydroxyacetone phosphate is converted into the glyceraldehyde 3-phosphate. So essentially you get two of these molecules at the end of the day, one from the splitting directly, and then one from the isomerization. All right. The second part are energy generating reactions. So reaction 6, oxidation, phosphorylation. So the aldehyde group of each glyceraldehyde 3-phosphate is oxidized to a carboxyl group by the enzyme NAD+, which is reduced to NADH and H+. A phosphate adds to the new carboxyl groups to form two high-energy molecules, or two molecules of the high-energy compound, 1,3-bisphosphate glycerate. So remember, NAD+ catalyzes the formation of carbon to oxygen double bonds, catalyzes sort of oxidation reactions. So the conversion of an aldehyde to a carboxylic acid is oxidation, okay? And NAD+ adds into that process. And then essentially the OH, the H portion of the carboxyl group is replaced by a phosphate group. Reaction 7, phosphate transfer. Phosphorylation transfers a phosphate group from each 1,3-bisphosphoglycerate to ADP, producing two molecules of ATP. At this point, two ATPs are produced, balancing the two ATPs consumed in reactions 1 and 3. Reaction 8, isomerization. 2,3-phosphoglycerate molecules isomerize, moving the phosphate group from carbon 3 to carbon 2, yielding two phosphoglycerates. Dehydration. Each phosphoglycerate undergoes dehydration, of course, which is the removal of water, to give two high-energy molecules of phosphoenol pyruvate. Where is the water removed from? We're taking off the hydrogen from here and the OH from here to give us this phosphoenol pyruvate, the En representing the carbon-to-carbon double bond that's present in that structure. And then reaction 10, phosphate transfer. In a second direct phosphate transfer, phosphate groups from 2-phosphoenol pyruvate are transferred to 2-ADP to form 2-pyruvate and 2-ATP. So here's one of the key points. At the end of the day, when glycolysis occurs and one glucose molecule enters the process, you get at the end of that 2 molecules of pyruvate and 2 molecules of ATP. So the one glucose at this point is going to give us 2 pyruvate and 2 ATP. And here's the structure of pyruvate. So in glycolysis, 2 ATP and add phosphate to glucose and fructose, 1,6-bisphosphate. 4 ATPs are produced during phosphate transfers. So there is a net gain of 2 ATP and 2 NADH when glucose is converted into 2 pyruvate. And also that generation of 2 NADH is going to be a source of energy as well. So that's also a key product. So 1 glucose, 2 pyruvates, 2 NADH, 2 ATP. That's the bottom line essentially of glycolysis. And this part is something that you should have in mind. Okay. Pyruvate pathway. So then what happens to the pyruvate? Aerobic and anaerobic. So pyruvate is converted to acetyl coenzyme A under aerobic conditions and to lactate under anaerobic conditions. Aerobic conditions. Under aerobic conditions, oxygen present, 3-carbon pyruvate is decarboxylated. Essentially, decarboxylation essentially refers to removal of a carboxyl group. Now here you have it in a deprotonated form, but essentially you still have removal of the carboxyl group. 2-carbon acetyl coenzyme A and carbon dioxide are produced. So 1 pyruvate is going to produce 1 acetyl coenzyme A, but essentially you have 2 pyruvate entering the process, and therefore you're going to get 2 molecules of acetyl coenzyme A. Not to worry about anaerobic conditions. All right, citric acid cycle. All right, so the citric acid cycle is a series of reactions that connects the intermediate acetyl coenzyme A from the metabolic pathways in stages 1 and 2 with electron transport and the synthesis of ATP in stage 3. As a central pathway in metabolism, the citric acid cycle uses the 2-carbon acetyl group in acetyl coenzyme A to produce CO2, NADH and H+, and FADH2. Is named for the citrate ion from citric acid, a tricarboxylic acid which forms in the first reaction. It's also known as the tricarboxylic acid cycle, TCA, or the Krebs cycle. In the citric acid cycle, an acetyl group in acetyl coenzyme A bonds to oxaloacetate, which has 4 carbons, to form citrate, which has 6 carbons. And we know the acetyl group has 2 carbons associated with it. 2 decarboxylation reactions remove carbon atoms as CO2 molecules to give succinyl coenzyme A, which has 4 carbons. A series of reactions converts 4-carbon succinyl coenzyme A to oxaloacetate, which combines with another acetyl coenzyme A, and the citric acid cycle starts all over. All right. So here's an outline of the citric acid cycle. And again, you don't have to know this entire diagram. You don't have to know it. All right, so we'll just go through it pretty quickly. So the acetyl group, 2 carbons from acetyl coenzyme A bonds with oxaloacetate, 4 carbons to yield citrate, 6 carbons. Citrate undergoes isomerization to yield its isomer isocitrate, which provides a secondary alcohol group that can be oxidized in the next reaction. Reaction 3, oxidation and decarboxylation. The secondary alcohol group in isocitrate is oxidized to a ketone and also produces hydrogen ions and electrons that reduce NAD+ to NADH and H+. A decarboxylation converts a carboxylate group to a CO2 molecule, producing alpha ketoglutarate, which has 5 carbons. Alpha ketoglutarate undergoes decarboxylation to produce a 4-carbon compound that combines with coenzyme A to form succinyl coenzyme A, hydrogen ions and electrons and also produces hydrogen ions and electrons that reduce NAD to NADH and H+. Reaction 5, hydrolysis. Succinyl coenzyme A undergoes hydrolysis to succinate and coenzyme A. The energy release is used to add a phosphate group to GDP, which is known as guanosine diphosphate, which yields GTP, guanosine triphosphate, a high energy compound similar to ATP. Reaction 6, oxidation. Hydrogen is removed from each of the 2 carbon atoms to succinate, producing fumarate, a compound with a trans carbon-to-carbon double bond, 2 hydrogens that are used to reduce the coenzyme FAD to FADH2. Reaction 7, hydration. A hydration reaction, the addition of water, to the double bond of fumarate yields malate, a secondary alcohol. Reaction 8, oxidation. The secondary alcohol group in malate is oxidized to a ketone, forming oxaloacetate, hydrogen ions, and electrons for the reduction of NAD+ to NADH and H+. So, the overall reaction. All right, so this is the important part. We can write an overall chemical reaction for one complete term for one complete turn of the citric acid cycle as follows. One molecule of acetyl coenzyme A enters the cycle, and you generate coenzyme A, 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of GTP. So 1 molecule of acetyl coenzyme A enters the citric acid cycle. You get 3 NADH, 1 FADH2, and 1 GTP. But remember, 1 glucose, 2 pyruvates, 2 pyruvates, 2 molecules of acetyl coenzyme A. So in total, 6 Na, and I always write sodium, capitalize that. 6 NADH, 2 FADH2, and 2 GTP. Keep that up. This is the important part. From my perspective. All right. Okay. Electron transport and oxidative phosphorylation. So, stage 3. From 1 glucose. In stage 3, for each glucose molecule that completes glycolysis. Right? So from 1 glucose, glycolysis gives us 2 ATP, 2 NADH. The oxidation of 2 pyruvates gives us 2 molecules of NADH. And then the citric acid cycle with 2 acetyl coenzyme A's give us 2 ATP, 6 NADH, and 2 FADH2. So you go back to glycolysis. Glycolysis gives us 2 molecules of ATP, 2 molecules of ATP, 2 molecules of NADH. Then the oxidation of 2 pyruvates gives us 2 molecules of NADH. And then the citric acid cycle with 2 molecules of with acetyl coenzyme A gives us 2 ATP, 6 NADH, and 2 FADH2. So from 1 glucose, we get 4 molecules of ATP, 10 molecules of NADH, and 2 molecules of FADH2. Electron transport. In electron transport, or the respiratory chain, hydrogen ions and electrons from NADH and FADH2 are passed from one electron carrier to the next and combine with oxygen to make water. Unfortunately, if you ask me to really break this all down for you, this is not one of my strong points. So again, I get the whole electron transport thing. Again, something you do not have to memorize, but just we'll go over quickly. The energy released during electron transport is used to synthesize ATP from ADP and PI, phosphate, a process called oxidative phosphorylation. As long as oxygen is available for the mitochondria, electron transport and oxidative phosphorylation function to produce most of the ATP in the cell. Mitochondria electron transport. So a mitochondria contains an outer membrane, an inner membrane, and a highly folded inner membrane. An outer membrane, an intermembrane space, and a highly folded inner membrane that surrounds the matrix. Enzymes and electron carriers along the inner membrane required for electron transport. 4 distinct protein complexes, complexes 1, 2, 3, and 4, located within these membranes. 2 electron carriers, coenzyme Q and cytochrome C, are not firmly attached to the membrane, function as mobile carriers shutting electrons between the protein complexes that are bound to the inner membrane. Electron carriers. Coenzymes NADH and FADH2 are oxidized in the enzyme complexes, providing electrons and hydrogen ions for ATP synthesis. Not going to go through all four complexes. Oxidative phosphorylation. In the chemosomatic model, each complex acts as a protein pump by using hydrogen ions from the oxidation of NADH and FADH2 out of the matrix and into the intermembrane space. The increase in proton concentration lowers the pH and creates an H+ or electrochemical gradient. To equalize the pH and charge between the intermembrane space and the matrix, the protons, the hydrogen ions return to the matrix by passing through a protein complex called ATP synthase. The process of oxidative phosphorylation couples the energy from electron transport to the synthesis of ATP from ADP. And this is probably the most important statement of all the complex stuff in the last couple slides. The process of oxidative phosphorylation couples the energy from electron transport to the synthesis of ATP from ADP. So this energy from electron transport through the generated from the process of oxidative phosphorylation combines ADP and phosphate to generate ATP. Okay. When NADH enters electron transport at complex 1, the energy released from its oxidation is used to synthesize 2.5 ATP. So 1 NADH, when it undergoes oxidation at complex 1, generates 2.5 ATP. FADH2, which enters electron transport at complex 2, which is at a lower energy level. Thus, FADH2 provides energy to produce 1.5 ATP. So 1 FADH2 is going to produce 1.5 ATP. In glycolysis, the oxidation of glucose stores energy in 2 NADH and 2 ATP. Glycolysis occurs in the cytoplasm, where hydrogen ions and electrons from NADH are transferred to compounds for transport into the mitochondria. In some cases, less than 5 ATP is produced from the 2 NADH from glycolysis because of the way these NADH molecules are transported from the cytosol to the mitochondrial matrix. To simplify our calculations of the ATP produced from glucose oxidation, we will not consider this potential reduction in ATP production. Because if you think about it, based on what we saw in the last slide, if you generate 2 NADH, and we saw that there are 2.5 ATP for every 1 NADH, well, that's going to give us 5 ATP. So that's where this number 5 comes into play. So glycolysis yields a total of 7 ATP. 5 of them come from the 2 NADH that are generated in the glycolysis process. And then you get 2 ATPs directly from the glycolysis process. So essentially, 1 glucose gives us 2 molecules of pyruvate, 2 ATP, and 2 NADH, or if we combine these two sort of together, 1 molecule of glucose, and again, this is just from glycolysis, gives us 2 pyruvate and 7 ATP. Of course, the 2 pyruvate move on. But at the end of glycolysis, we already have 7 molecules of ATP. ATP from the oxidation of 2 pyruvates. So under aerobic conditions, two pyruvates enter the mitochondria and are oxidized to 2-acetyl coenzyme A, CO2, and 2-NADH. 2-NADH enter electron transport to provide 5-ATP. So, again, we saw one molecule of NADH, 2.5 ATP. If you have two molecules of NADH that are being generated, that's going to give you 5 ATP. So instead of saying here 2 NADH, instead they're telling us 5 ATP. So far we've got 7 and we've got 5. So far we've got 12. Before we get to the citric acid cycle. Because the two molecules of acetyl coenzyme A are then going to move on. Move on to what? Move on to the citric acid cycle. All right. So one turn of the citric acid cycle provides 3 NADH, which provides a total of 7.5 ATP. One molecule of FADH2, which provides 1.5 ATP. Because remember, as we saw earlier, one molecule of FADH2 is going to undergo oxidation to produce 1.5 ATP. And then finally, I mentioned it earlier, I talked about the GTP. The GTP is a source of energy as well. One GTP will actually produce 1 ATP. So if you add those up, one turn of the citric acid cycle is going to give us 10 ATP. But remember, how many molecules of acetyl coenzyme A enter the citric acid cycle? 2. So there are 2 turns of the citric acid cycle. So the citric acid cycle produces 20 ATP. So far we've got 7 and 5 and 20, 32 ATP. Okay. So ATP produced by the complete oxidation of glucose. 5 and 2 is 7. Glycolysis. Oxidation and decarboxylation, 5. And I would remember these different parts. This table here is probably something I would know. So oxidation of glyceraldehyde 3-phosphate gives 2 NADH and 5 ATP. Direct phosphorylation 2-triose gives 2 ATP. The 2-pyruvate, the 2-acetyl coenzyme A, 2 NADH and 5 ATP total. And then here, the citric acid cycle, I would probably remember oxidation of 2-isocitrate gives you 2 NADH. Oxidation of 2-alpha-ketoglutarate gives you 2 NADH. 2 direct phosphate transfers, 2 GTP. Oxidation of 2-succinate, 2 FADH. Oxidation of 2 malates, 2 NADH. And then the corresponding ATP numbers. Add those all up. One molecule of glucose is going to generate 32 molecules of ATP. This table here is probably something that I would remember. So the complete oxidation of glucose to carbon dioxide and water yields a total of 32 ATP. And this slide here gives you a little bit about where, I don't particularly love this picture. I like this breakdown of this table a lot better. Let's see. Almost just about out of time. All right, we'll just go through some of this a little bit quickly. So degradation of amino acids. Once the carbon, so let's go. Degradation of amino acids. So proteins provide energy when carbohydrate and lipid resources are not available. The carbon atoms from amino acids are used in the citric acid cycle for the synthesis of fatty acids, ketone bodies, and glucose. Most of the amino groups are converted to urea. Transamination. In transamination, amino acids are degraded in the liver. An amino group is transferred from an amino acid to an alpha-keto acid, usually an alpha-ketoglutarate. A new amino acid and an alpha-keto acid are formed. When alanine combines with alpha-ketoglutarate, pyruvate and glutamate are produced. Okay, I would just know a little bit generally about the definition of transamination. So here's a transamination reaction: alanine with alpha-ketoglutarate, giving you pyruvate and glutamate. All right. So an amino group is transferred from amino acid to an alpha-keto acid, usually alpha-ketoglutarate. Okay. So here's the amino group in alanine that's being transferred. So it's taking the place of the oxygen in the alpha-ketoglutarate to give you glutamate, and then the oxygen ends up with a C double bond O group here to give you pyruvate. So that's essentially a good example of a transamination reaction. Oxidative deamination. So oxidative deamination removes the ammonium group, NH3+, from glutamate as an ammonium ion. NH4+ provides hydrogens for the NAD+ coenzyme, regenerates alpha-ketoglutarate, which can enter transamination with an amino acid. The urea cycle removes toxic ammonium ions from amino acid degradation, converts ammonium ions to urea in the liver, okay, produces 25 to 30 grams of urea daily for excretion in the urine. ATP energy from amino acids. So carbon skeletons of amino acids form intermediates of the citric acid cycle, produce energy, enter the citric acid cycle at different places depending on the amino acid. 3 carbon skeletons, such as alanine, serine, and cysteine, get converted to pyruvate. 4 carbon skeletons, such as aspartate and asparagine, get converted into oxaloacetate. And 5 carbon skeletons, such as glutamine, glutamate, proline, arginine, and histidine, get converted to glutamate. And there's just a little bit of the intermediates of the citric acid cycle from amino acids. And I think cell membranes, fluid mosaic model. Yeah, we'll just touch on this quick. So cell membranes are semi-impermeable so that nutrients can enter the cell and waste products can leave. So that nutrients can enter the cell and waste products can leave. Separate cellular contents from the external environment. Consist of a lipid bilayer made up of 2 rows of phospholipids. Have an inner portion made of the nonpolar tails of phospholipids, with the polar heads, which of course is the phosphate and the amino alcohol groups, at the outer and inner surfaces. In the fluid mosaic model of a cell membrane, proteins and cholesterol, and why do I think? Because it's the wrong chapter. Sorry about that. That might be it. Yep. I think that's it. That was it. Sorry. I just saw chapter 17 on there, and that was at the end of chapter 17. All right, and that's it. Appreciate watching the videos, and hopefully they were a help during the semester, and wish you best of luck on the final quiz.
Great, thank you.
You're welcome. Good luck. Thank you. Take care.